This is a video to help you understand the mechanisms of transcription. And it all starts with the simplest of rules. And that rule is that in DNA, the nucleotide A always binds with T with a weak hydrogen bond. G always binds with C. In this relationship, this pairing of A to T and G to C, this is known as complementary bonding. And it's on this simple rule that all of molecular biology and all of evolution, inheritance, cell division, it's based on this one simple rule. Everything stems from this. And so now once I have this rule established, I can now start thinking about the structure of DNA. How does DNA manage itself? And as you remember, the recipe for proteins is when carried on DNA, and that recipe is spelled out in nucleotides, in groups of these 3 nucleotides together called a codon. So each group of 3 nucleotides is called a codon. Each codon specifies for a particular amino acid in the protein sequence. So in the DNA that I'm making here, my first codon is going to have the sequence ATG. It happens to specify for the amino acid methionine. The second codon I'm going to give it is CCC. The third codon AAA. And I'm going to insert a stop codon which has the sequence TAA. So now I have a recipe for a protein that I want to make here. And because I have this simple rule established, my complementary rule, I can now know what the complementary strand of my DNA molecule is going to be. I follow my same rules. A binds with T, T binds with A, G binds with C. C, no problem. I'm going to get G, G, G. A is going to get T, T, T. T complements A complements T. And now this strand here, the top strand, this is the strand that carries the actual recipe for the protein. So this is going to be termed the sense strand. And the complementary strand down here is going to be called the antisense strand. And now DNA is really good at storing the information, but it's really crummy at making protein. So in order to make the protein, I need to use an intermediary, and that intermediary molecule is going to be called messenger RNA. It's another nucleotide type. And now the question is, how do I make an exact replica of the recipe that I need if all I have to work with is my complementary binding rule? And the answer is that I'm going to use the antisense strand as the template, and in so doing I'm going to make a replica of the sense strand. Now the only thing you have to remember about RNA is that instead of using T, it uses U, otherwise it's the same, and that U actually confers a bit of instability to the RNA, but we can just remember that. So in order to demonstrate how this works, let's separate off the strands here so I can pull apart the DNA antisense strand here, just to make it a little bit easier to work with here. And now I'm going to pull this down. And now what I'm going to do is I'm going to make my messenger RNA using my pairing rule, use my complementary rule. So here, my T makes a messenger RNA A, because remember, I don't use a T, I use a U instead. RNA is just weird like that, uses a U. C becomes G. Now my G's, the complement of that is CCC. The complement of the T's is going to be AAA. And here, A now becomes U, and T becomes A. And so now I have my messenger RNA strand here. And when I compare my messenger RNA strand with my sense strand of DNA, you can see that it's a replica. AUG, it uses U instead of T. The C's are replicated, the A's are replicated in here. Instead of using a T, it's got a U. But in other words, as long as I understand that I've substituted U for a T in my messenger RNA, I have a perfect replica of the sense recipe. And now I can send this messenger RNA off to the protein factory, the ribosome, and I can now start to make the protein. So let's move this out of the way just a bit to make some room here. And now what I'm going to do is I'm going to clip off the messenger RNA part here. Just take this one part here. And when I do that, I bring it down here. And now what I want to do is show you how the ribosome is going to assemble this. So in order to assemble this, the transfer RNA is going to be what's going to bring the RNA, bring the amino acid to the factory here. And the transfer RNAs have complementary sequences to this messenger RNA sequence here. So the complementary sequence, the AUG, is going to be U, A, C. And that happens to be bound to a transfer RNA. Remember, that's got that 4-leaf clover shape here. And that's always bound to the amino acid methionine. So it's going to be this transfer RNA that is now moved to the ribosomal factory here. And now it's going to be put into place here, specifying that that's where the first amino acid is going to be. Now when I look for the complement of CCC, that's going to have an anticodon of GGG. Again, the ribosome has that same motif, the same shape, but now the GGGs, they are always bound to the amino acid proline. And so now, that transfer RNA is brought into place here. Thus, and when I look for AAA, now that anticodon is TTT. And this transfer RNA, the TTT, oh, it's not TTT. My mistake here. Because it's RNA, right? We're not going to use T. It's going to be UUU. So we have U, UU, and that UUU is always going to be bound to the amino acid lysine. Okay, now I can bring this into place here. Now I look for my final sequence, the UAA, and that is going to be bound to the anticodon, which happens to be ATT, or AUU in this case, because it's RNA, right? And this is bound to the stop codon. So now when this codon is brought into place, what we see is that now it commands the factory to stop, so that the amino acid produced is going to be methionine, protein, lysine. The stop codon comes, kicks this protein off the ribosome, and that's going to be my finished product here. Okay, so that's all well and good. Now, what we need to do next is to figure out how to talk about this stuff. How do we discussed this in terms of what is it going to look like on our lab practical. What is that problem going to look like? And as we've seen, that problem is going to look like a codon table here. So let me get my table. And now when I get my codon table, here is the problem I'm going to be looking at. And so now what I want to see is how I'm going to fill this table. So in this particular case, I have the sense DNA given to me, ATG, and I have the messenger RNA, AUG. That makes sense because it's a replica of this, except instead of having a T, I have a U. So I accept that. That's good. Now the question is, what is my antisense DNA going to look like? So now I'm just going to make a complementary copy of the sense DNA. So in this case, if I make a complementary copy of the ATG, then I'm still DNA, right? So it's going to become T. T's become A, and the G becomes a C. So that's the sense, the antisense messenger RNA. And then I want to know what is going to be my amino acid sequence or my amino acid choice in this particular problem. So now I need my RNA codon table. So let me get my RNA codon table. So here's my RNA codon table. And now what I can do is I can look up exactly what is going to be the amino acid specified by the codon AUG. So here's A, I find my U, and I find my G, and it says methionine. So then the correct amino acid choice here is going to be methionine. Also the start codon of my protein. Now look here in my next column, all I'm given is messenger RNA, and the messenger RNA is going to be AAG. So let's make it simple. What must the sense DNA be? It must be a replica of this, and since I only have A's and G's, a replica of the sense DNA here. This also has to be AAG because I'm making a faithful copy here. No T's are involved, so it's going to be the same. What is going to be the antisense DNA here? Now I use my complementary rule. A becomes T. I'm still in DNA, so I'm using T, 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 and now C. And I want to say, what is the amino acid specified by AAG? Here's A, G. The amino acid is lysine, so this answer here is going to be lysine. Here in my final column, I just have sense DNA, and I have nothing else to go on. So if I have sense DNA, what is my antisense DNA? I make a simple, follow my simple complementary rule. T becomes A becomes T. Now what is going to be messenger RNA? I'm going to make a complementary of my antisense DNA. And so A, it's RNA, so it becomes U. The T becomes A. And if I look at my codon table, UAA is going to specify for a stop codon. So that's all there is to filling out the table and understanding how these relationships between the different sense strand of DNA, the antisense DNA strand, messenger RNA, and how that message is successfully translated into protein.